Hippocrates famously said, "All disease starts in the gut," more than 2,000 years ago. In this series, we will be discussing the common causes of leaky gut and how to break the cycle of inflammation and keep your organs and body healthier and stronger for longer. Understanding the structure of the gut barrier can help with formulating precision medicine treatment approaches to leaky gut. Through this process, you'll also learn about nutrients, nutraceuticals, and interventions that can get to the root of inflammation and help repair the gut barrier.
Before we can talk about what leaky gut is and why it occurs, it's important to have a discussion about the anatomy of the gut lining, and then that will make it drastically easier to come up with solutions to heal leaky gut and to understand how to address the root cause. So first, we're going to have a discussion of what the gut lining looks like. We're going to talk about the major components in the different regions of the gut layers, and we're going to talk about the segments such as the small intestine and the large intestine and some major differences between them anatomically. Then we're going to get into a discussion of the cell types, because there are different cell types in the intestinal epithelial cell layer, and what these different cell types do, how they function, and then the basic receptors that can pick up on signals from the gut lumen and how certain molecules are transported or internalized and destroyed, depending on whether we're talking about nutrient absorption or the immune response. Next, we're going to talk about transport in a little bit more detail and the ways that molecules can be transported across the gut barrier, including through or between the cells. And then finally, we're going to take a closer look at the transport between the cells, which is called paracellular transport, and the major proteins that are involved in that process. So let's get started.
What exactly is leaky gut or intestinal hyperpermeability? Before we get into that, it's important to know that the function of the gut is to absorb nutrients, ions, and beneficial molecules. The gut is made up of a one-cell layer thick barrier of intestinal epithelial cells. These epithelial cells are connected by tight junctions, which are proteins that help the cells adhere together, like brick and mortar. Tight junctions also vet which molecules can pass through and get absorbed into the bloodstream. Not only do tight junctions provide a physical fence, but they also offer a chemical fence. More on that later. Normally, tight junction proteins line epithelial cells of the gut, acting like door locks, keeping the contents of the gut outside and the body's internal milieu inside. However, sometimes molecular signals in the gut, whether from diet, microbes or microbial products, chemical exposures, and so on, can signal those tight junctions to break apart. That causes intestinal epithelial cells to come apart, and the contents of the gut are allowed to enter systemic circulation. Once foreign molecules enter the bloodstream, they can potentially access any organ and trigger systemic inflammation. So, all in all, healthy functioning of the gut lining is crucial for a well-adapted body, from digestion and absorption to immune function and brain health. So what exactly is going on during leaky gut? What is the mechanism? How does it work? And what is the anatomy of the gut barrier? So for gut contents to leak outside the gut wall into the body, they need to pass through three major layers: the mucus, the intestinal epithelial cell or IEC layer, and the underlying immune defense mechanisms. The lumen is the space occupied by food contents and microbes. There is a certain level of defense against leaky gut even at the level of the lumen. Here, bacteria and antigens are degraded by bile, gastric acid, and pancreatic juice. In addition, commensal bacteria inhibit colonization of pathogens by producing antimicrobial substances. Beneath the lumen is the mucus layer. Mucosa are also present at the airways, oral cavity, digestive tract, genitourinary tract, and skin. Mucus is full of antimicrobial compounds. Mucus itself prevents bacterial adhesion. Gut mucus, in particular, helps with digestion, nutrient absorption, waste secretion, and keeping the gut relatively alkaline. The small intestines digest food and absorb nutrients, while the large intestines absorb water and electrolytes. Bacterial density is very high in the large intestines, necessitating a thick inner mucus layer full of antimicrobial compounds and immunoglobulins. This layer is normally impenetrable to bacteria. Compounds in the mucus, like mucins, prevent large particles, including most bacteria, from directly contacting the epithelial cell layer. In addition to that, B cells and plasma cells in the lamina propria, which is the underlying immune tissue, which we'll get to later, as well as goblet cells, which are in the intestinal epithelial cell layer, secrete IgA, which binds to specific bacteria, bacterial products, invading microorganisms, and toxins, and it entraps them in a mucus layer, neutralizing them. We'll get to goblet cells and talk more about them in just a little bit. Over time, the thick mucus layer turns over, just like skin, and it thins out. This thinner mucus can serve as a nest for the microbes, while our intestinal epithelial cells are continually working to produce thick mucus. Intestinal epithelial cells. Intestinal epithelial cells, or enterocytes, comprise the single-cell thick internal lining of the gut. This layer is composed of different types of specialized intestinal epithelial cells. This includes absorptive enterocytes, goblet cells, tuft cells, enteroendocrine cells, Paneth cells, and microfold or M cells. IECs are rapidly renewed and replaced every couple days. These cells work to maintain barrier integrity. IECs form a brush border at their apical surface to keep things moving. Many intestinal epithelial cells adhering together form villi and crypts. Pore size is relatively smaller at the villus tip and larger at the base of the crypt. As a result, size selectivity varies. The permeability is higher in the crypt and lower at the villus. IECs also serve immune functions. IECs can react to noxious stimuli by secreting chloride and antimicrobial peptides, such as beta-defensins and Reg3. They recognize prokaryotic-associated molecular patterns, or PAMPs, with toll-like receptors, or TLRs, that are expressed on their cell surface, as well as nucleotide-binding oligomerization domain, or NOD-like receptors, which are expressed in the cytoplasm. These receptors activate defense mechanisms by secretion of antimicrobial peptides. IECs can also phagocytose bacteria and sequester and neutralize bacteria and toxins, and they communicate with underlying immune cells to regulate inflammatory response against bacterial toxins. Absorptive enterocytes are the most abundant epithelial cells. They do as their name sounds, absorb nutrients. Goblet cells secrete mucins, among which mucin 2 is the most abundant, which prevents adhesion of bacteria on epithelial cells. Goblet cells also secrete molecules that are central to both defense and repair of the epithelial layer, including IgA, mucus, trefoil peptides, and resistin-like molecule beta. Tuft cells are more rare and less well characterized, but they are essential for immunity against parasitic helminths. In response to helminth infection, these cells orchestrate the type 2 immune response by producing IL-25, which activates ILC2 or innate lymphoid cell type 2 cells. ILC2 cells can in turn recruit eosinophils to protect the intestinal epithelium. Enteroendocrine cells produce and release hormones into the submucosal space to interact with neurons or immune cells. These hormones include substance P, bradykinin, prostanoids, and in response to various mechanical, chemical, and nervous stimuli, most of the serotonin in the gut. Serotonin stimulates motility and secretion in response to most injurious substances to move them down towards the less permeable colon. It also secretes glucagon-like peptide 2, GLP-2, that is trophic to small bowel epithelium and induces healing. Paneth cells are numerous in crypts and establish a chemical barrier by secreting antimicrobial peptides. When exposed to bacteria or bacterial products such as LPS, lipopolysaccharide, they produce antimicrobial peptides, including lectins, alpha-defensins, Reg3 proteins, and lysozyme to keep microbes at bay. Microfold cells, or M cells, are critical for the function of mucosal IgA responses. They secrete IgA, which helps present bacterial antigens to dendritic cells. They reside in the follicle-associated epithelium, or FAE, above Peyer's patches, and are found in low levels, comprising around 5% of the FAE. M cells are specialized for the uptake of intestinal antigens, including live bacteria. These bacterial antigens bind to receptors, and then they are transported through transcytosis through the cell to underlying lymphoid tissues, where they are retained and destroyed. Next up, we have gut-associated lymphoid tissue, or GALT. GALT is made up of the muscular mucosa, what the whole GALT sits on, as well as Peyer's patches, lymph nodes, and lamina propria. Peyer's patches are the most important sites for the induction of T-cell-dependent IgA class switch recombination. Human young adults have over 200 Peyer's patches in their small intestine. They lack lymphatic vessels. Lamina propria contains innate and adaptive or acquired immune cells, particularly dendritic cells, plasma cells, macrophages, lymphocytes, and in some cases, neutrophils. These immune cells secrete chemical substances including IgA, cytokines, chemokines, and mast cell proteases. Plasma cells secrete secretory IgA, or SIG-IgA, into the gut lumen. B lymphocytes secrete cytokines, chemokines, and anti-inflammatory IL-22. Mast cells secrete proteases, which can mobilize inflammatory responses and clearance of bacterial pathogens. Dendritic cells sample the gut lumen by extending dendritic processes across tight junctions in the distal small intestine. Dendritic cells can be conditioned by TGF-beta and retinoic acid, both of which can enhance T regulatory cell differentiation. Immune tissue also includes intraepithelial lymphocytes within the intestinal epithelial cell layer near the basal side. They are located above basement membrane, but subjacent to tight junctions. Additionally, M cells, which are part of that mucosa-associated lymphoid tissue, or MALT, are specialized antigen-presenting epithelial cells, as we mentioned before. They internalize antigens from the gut lumen into vesicles and transport them transepithelially directly to intraepithelial lymphocytes and to subepithelial lymphoid tissues. Minor barrier defects allow bacterial products and dietary antigens to cross the epithelium and enter the lamina propria. This can lead to disease or homeostasis. If the foreign materials are taken up by antigen-presenting cells, such as dendritic cells, those direct the differentiation of T helper 1, Th1, or Th2 cells, and then disease can develop. In this process, antigen-presenting cells and Th1 cells can release tumor necrosis factor and interferon gamma, which signal to epithelial cells to increase flux across the tight junction leak pathway, thereby allowing further leakage of bacterial products and dietary antigens from the lumen into the lamina propria and amplifying the cycle of inflammation. This may ultimately culminate in established disease. Alternatively, interleukin-13, IL-13, released by Th2 cells, increases flux across small cation-selective pores, potentially contributing to ongoing disease as well. Conversely, homeostasis may dominate if antigen-presenting cells promote regulatory T cell differentiation, also known as Tregs, which can be enhanced by epithelial cell-derived transforming growth factor beta, or TGF-beta, as well as retinoic acid. The Treg cells display latency-associated peptide, or LAP, on their surfaces and may secrete IL-10 and TGF-beta to prevent disease. Leaky gut causes bacteria to translocate and causes expansion of CD4-positive T cells and IL-17A production that limits enteric pathogen invasion. The final layer is the enteric nervous system. This layer activates endocrine and secretomotor mechanisms and mediates intestinal propulsive motility. It also secretes some transmitters, including serotonin and histamine. Serotonin signaling can help bad things move along so they don't get absorbed. As you can see, the intestinal epithelial cells are situated between the mucus and the underlying immune system, and they play a major role in gut barrier integrity. Now let's circle back around to intestinal epithelial cells and take a closer look at how molecules are transported and what goes wrong in leaky gut. Molecules can cross between cells, which is called paracellular transport, or through cells, which is called transcellular transport. Transcellular pathways can involve passive diffusion across the cell membrane or can be carrier or receptor mediated. Meanwhile, the paracellular pathway involves intramembrane passive diffusion between spaces through adjacent cells, allowing ions and solutes through, but preventing proteins, lipids, and microbial-derived peptides from passing through. All right, first let's talk about transcellular pathways. Transcellular transport allows the passage of bacterial or food antigens across cell membranes. There are a few basic kinds of transcellular transport: passive transport, passive diffusion by efflux pumps, active transport, and endocytosis. In passive transport, nonpolar compounds like soluble lipids pass through the phospholipid bilayer by simple diffusion. Through passive diffusion by efflux pumps, small hydrophilic compounds and nutrients mainly use membrane channels and transporters. Active transport is carrier-mediated and involves binding of a target molecule to the receptor of the transporter. The carrier undergoes a conformational change and the compound translocates. Finally, endocytosis occurs for larger molecules, such as proteins and bacterial products. Proteins or peptides bind to specific cell surface receptors, resulting in transcytosis. Endocytosis functions in antigen surveillance in the GI tract. Immunoglobulins and viruses undergo clathrin-mediated endocytosis. Food allergens complex with IgE and bind to CD23 on the apical side. One particular type of endocytosis, called phagocytosis, involves toll-like receptor-mediated internalization of gram-negative bacteria in enterocytes. Through this process, bacteria, viruses, and particles bind receptors. Finally, micropinocytosis is nonspecific and internalizes extracellular fluids, dissolved molecules, viruses, and apoptotic cell fragments. Lipid rafts, or caveolae, mediate internalization of some enterotoxins and viruses into enterocytes. This involves invagination of cholesterol-rich areas of plasma membrane that contain a coating protein called caveolin. Now let's talk about paracellular pathways. In the paracellular pathways, there are three basic options: transport, leakage, or apoptosis, or unregulated permeability. The leak pathway allows paracellular transport of large solutes, including limited flux of proteins and bacterial lipopolysaccharides. The size limit hasn't been defined precisely, but research indicates that whole bacteria are too large to pass. There is no charge selectivity for this pathway. TNF-induced myosin light chain kinase activation, or MLCK, seems to increase paracellular flux through the leak pathway. Through formal transport, tight junction-associated claudins define small pores that are the primary determinants of charge selectivity. They exclude molecules larger than 4 angstroms. Expression of claudins can vary by organ and by region within organ, and is modified by external stimuli like cytokines. Finally, there is the apoptotic pathway. This pathway is completely unregulated. Extensive apoptosis of epithelial cells is a problem. However, the relevance of single-cell apoptosis to barrier dysfunction remains controversial, as different results are obtained in diverse experimental systems. Paracellular transport is mainly what we're talking about when we're talking about leaky gut. So let's take a closer look at that paracellular pathway and which molecules are transported. Under physiological conditions, only water and solutes, like electrolytes, can cross the epithelium through the paracellular pathway. The junctional complex of IECs involved in the paracellular pathway includes various proteins. Broadly, the paracellular pathway is composed of the apical junctional complex, which includes tight junctions and adherens junctions. It also includes gap junctions and desmosomes. Tight junctions are the principal regulators of paracellular permeability. They promote the structural integrity of epithelial cells lining the gut and seal the paracellular spaces between epithelial cells. Tight junctions involve various proteins, including claudins, occludin, junctional adhesion molecule A or JAM-A, intracellular scaffolding proteins such as zonula occludins, and cytoplasmic proteins, which help coordinate cytoskeletal dynamics. Tight junctions limit the transport of hydrophilic molecules. They regulate size and charge selectivity and allow some medium-sized hydrophilic molecules, ions, or positively charged molecules to pass through. They prevent transport of proteins, such as antigenic macromolecules, lipids, and microbial-derived peptides. They're responsible for water, certain small molecules, and ions passing through the barrier. Next, the adherens junction plays a role in the stabilization of cell-cell contacts. Finally, desmosomes are located under the apical junctional complex and join adjacent cells together and provide anchoring sites for intermediate filaments. When we're talking about treatment approaches to leaky gut, we're getting straight to the heart of the issue. Our goal is fortifying those paracellular tight junctions in those intestinal epithelial cells. We're also improving function of individual types of IECs, like goblet and Paneth cells, which are constantly at work absorbing nutrients, surveilling the gut to make sure our own microbiome doesn't get too close to the barrier, and destroying pathogens. And if you're interested in healing and nutraceutical approaches for improving intestinal integrity, please remember to subscribe as we're covering that in this series. In addition, we would love your comments. Any kind of engagement helps our channel out so much as we're just starting out. Once again, I appreciate you for being here and stay tuned for our next video on diseases associated with leaky gut and the relationship of leaky gut to aging. You won't want to miss this one. Thanks for watching. See you next time.